Can you hear me? Okay, thank you. Did you tell the other students to come, please? Did you tell the other students to come? Hello, hello, good morning. Did you tell the other students to come? Would you please tell the others if you know them? Or inform them in the group, please. Thank you, Fatima. So just wait a few seconds for them to come, okay? Okay, hello again. So many students doesn't come to the class. Why? Did you tell them? Okay, anyway, we start the session. So, Do you have any question from the last session? You told them? Thank you, Fatima. Do you have any? Okay, thank you. So let's talk about the achieving of the right balance between the culture age and inoculum size when preparing a sample of a non-lysogenic phage. What does it mean? Just listen to me. Just listen to me. If we want to culture the bacteria which are infected with the phage, what will happen? When should we capture the more phage? Look at here. The bacterial growth. We have a bacterial growth table. Look at this. If we want to show the bacterial growth, we have lag, log, and stationary. Do you remember? Bacterial growth, lag, log, logarithmic, and stationary phase of the bacterial growth. Do you remember? Okay. At first, our bacteria grows very slowly. At first, the bacteria grows very slowly. No problem, Mr. Ali. Look at here. At the phase of lag, the bacteria grows very slowly. So here, look at here. Culture density is low. So the bacteria are low. And if we add the phage to the bacteria, what will happen? Because the amount of a bacteria are low and the density is low, what will happen? All the bacteria will kill. Look at here, all the cells will kill. If we add the bacteria here, it means if they add the bacteria at the stationary phase, which culture density is too high, so we have so many bacteria. If we add phage here, culture is never completely lyses. So what do we have? We have low phage titer. So if we add the phage here, when the bacteria growth is low, when the density is low, what will happen? If we add phage here, the phages will destroy or lyse all the cells rapidly. So what do we have? We have low phage titer. Do we want this? Is it good for us? Low phage titers? We want more phages. Yes? So if we add the phages in this phase, which we have little amount of bacteria, all the bacteria will die. So we have low phages. Do you want this or no? No, so we don't want low phage titer. Yes? So listen here. If we have so many bacteria, for example, if the bacteria growth phase is here, so many bacteria, So the density is too high. If we add the phages here, what will happen? The culture is never completely lysed because so many bacteria we have here. So we have low phage titers. So do we want this? Tell me. Is it okay for us? No. Here, look at here. If we add bacteria which are here, it means if they are in logarithm, in log phase, the culture density is just right. If we add the bacteria which is here in log phase, so the bacteria is high, but there's not too much. If we add the phages, the culture continues to grow, but is eventually lysed. So what do we have? We will have high phage titers. So is it good? Yes, it's very good for us. So where should we add the phages to the bacteria? Here. In the log phase. In the log phase. This is the stationary. And this is the lag. Where do we want? Where do we want to add the phages? Here. Understand up to now? Okay, thank you. So, if we want to collect the phage particles, how can we collect the phage particles? Look at the picture. As you know, as I told you before, let's go to the previous slides. Let's go here. Okay. Do you remember this slide? I told you if we want to separate the bacteria from the phage, what should we do? We should centrifuge the culture. So the bacteria go down. Why? Why the bacteria go down? I told you because they are heavier than the phage. Yes? So because the bacteria are heavy, thank you, Fatima, they go down and we have phage in the supernatant. But how can we separate the phage? Listen to me. We can add polyethylene glycol. What does polyethylene glycol? Let me write it for you. Polyethylene glycol. Sorry, let me type it for you. Polyethylene glycol. Polyethylene glycol is PG. What is polyethylene glycol? Polyethylene glycol is a long-chain polymerase compound. Long-chain polymerase compound, which, in the presence of salt, absorbs water, thereby causing macromolecular assemblies such as phage particles can attach to them, and after we centrifuge the solution, we centrifuge the solution, because they are heavy, they can go down and they can be precipitated. So the precipitate can then be collected by centrifuge and redissolve in suitably small volume. Look at here. Look at here. Thank you, Mr. Haidar. This is phage suspension. Phage suspension. This is phage suspension. If we add polyethylene glycol is a long-chain polymer, I told you, in the presence of salt, which is NaCl, it will absorb the phage particles, and if we centrifuge the suspension, what will happen? The phage will precipitate down, will go guard the supernatant, and we can dissolve the plate. Do you understand? Okay, thank you. So, phages are very small particles. If we centrifuge, we can't have them in the plate because they are so small, so small and so light. If we centrifuge, we can't precipitate them. So we should add polyethylene glycol by the presence of salt can absorb this particle, absorb water, absorb this particle, become heavy, and when we centrifuge, they will go down. We discard the supernatant and resolve the phage particles. Do you have any questions up to now? So now we have phage particles. What do we have in phage particles? Can you tell me, please? What do we have in phage particles? What do we have in phage particles? Can you tell me? Do you remember, as I told you about the phage, for example, lambda? This was the capsid. Do you remember? Head and tail structure. And in the middle, we had what? We had genome. So what do we have? We have proteins, which are capsids, and we have genome, which are DNA. Yes? So we have DNA and proteins. DNA. Yes, of course. And we have capsid here too. Yes? So we should get rid of the capsid proteins. We want to purify the phage. So listen to me. If if we want to purify the phage, we can take it to cesium chloride density gradient centrifuge. Do you remember the last session I told you about cesium chloride density gradient centrifuge? This is the same. I told you cesium chloride density gradient, if we put the phage particles here, they usually density is about 1.45 to 1.50. So this is, we can have the DNA of the phages. So after that, we can extract it with phenol. We want to destroy the, here, let me see you. After this, we want to, this is phage particles, we want to remove the proteins. How can we remove the proteins? Can anybody tell me? Do you remember phenyl chloroform? As I told you, do you remember? We can extract and we can purify the proteins with phenyl chloroform. Do you remember? Yes. Okay. After that, we can add phenyl chloroform. Thank you, Fatima. Thank you, Fatima. Yes. We can add phenyl chloroform in the portion of 1,1, and after that we centrifuge it. We have two phases, and in the liquid aqueous phase we have DNA, so we can get rid of the proteins. Okay up to now? Thank you, Hale. Or we have another way. What is the another way to get rid of the proteins? We can add protease. For example, proteinase K. If we add proteinase K, what will happen? In proteinase K, we can get rid of the proteins. Understand? So we can take it in cesium chloride density. In this density, we can have phage particles, but in phage particles, we do have some proteins. We want to purify the DNA from the proteins. We can use phenyl chloroform, as I told you before, or we can use proteinase K. Okay, thank you so much. If you don't have any question, let's go to talk about purification of M13 phage. M13 phage. Do you remember what does the structure of M13? As you remember, it was filamentous. Do you remember? It was filamentous. How about the capsid? The capsid of like this, and a structure was what? Filamentous. Do you remember? What was the very important difference Very important difference between Landophage and M13. Can you please tell me what was the most important difference? Do you know? Do not kill the bacteria. Yes, thank you, Fatima. Of course, one of the most important difference was M13 never kills the bacteria, just slow down the proliferation. And what else? Fatima, can you tell me, please? What else? Okay. The second was its DNA can be single strand DNA and can be double strand circular DNA. Do you remember? If we have double strand circular DNA, we call it RF form, replicative form. So we have two kinds of DNA. One, single strand DNA, and the second one is double strand DNA. So if we have the replicative form, RF form of M13 can behave like a high copy number of plasmid. So we can very easily purify the double strand DNA plasmid from like, for example, like we extract the plasmid. How could we extract the plasmids? Let's go before. Let's go here. In the ethidium bromide chloride cesium density gradient. So if we have double strand form of DNA of M13, which we called RF form, how can we extract them? Because this is like the plasmid, we can treat it like the plasmid. How can we extract the plasmid? We could use ethidium bromide, cesium chloride density. So we can use it like, we can extract it like this. Take it to the cesium chloride, ethidium bromide density. We have RNA here, protein here, linear open circular here, supercoil here. What do we want? This part. Understand? Yes, okay. But do we just have double strand DNA for M13? No, we have single strand too. Yes, am I right? So we have single strand and we have double strand. So we have single strand in our medium, in our extracellular culture too. Okay, so look at the picture, please. Look at the picture. Here is culture of infected cells. Look at here. This is culture of infected cells. If we centrifuge the culture of infected cells, what do we have? Please tell me. What is heavier? What is heavier? What is heavier in this solution? No, no, no. What is heavier, which present plate? Do you understand my question? I told you here in this culture we have bacteria and phage. Okay, we centrifuge, and which is heavier? Which part are bacteria are heavier than the phage? Yes, bacteria are heavy. Yes, thank you. I just want to say آفرین. Did I tell you what does آفرین means? Let me write it for you. آفرین means very good. Very good. I think in Arabic it's مرحبا, huh? Bravo. أحسنت. Yes. We use أحسنت too. But آفرین is some usual form. So, آفرین فاطمه. Listen to me. If we centrifuge the solution, if we centrifuge the culture, because the bacteria are so heavy, they precipitate, they go down, they form plate, and we have M13 phage in the upper side, in the upper solution. Why? Because they are so light. This is heavy, go down, and this is so light. So, what do we want? We want the supernatons, we want the upper side, and remove it to another microtube, and we want to precipitate M13 phage. How could we precipitate M13 phage? How could we precipitate phage? I told you, if I centrifuge our solution, because the phage particles are so small, they cannot precipitate. I should add something else. To make it heavy and centrifuge. What was that? Polyethylene glycol. Thank you, Mr. Natek. Polyethylene glycol. So I should add. No. Polyethylene glycol. I want to precipitate M13. I told you phages are so small and so light. We should add polyethylene glycol. I told you polyethylene glycol by the site of salt, for example, NaCl. Absorb water and attach to M13 phage, and if we centrifuge, it goes down. So we centrifuge, we add polyethylene glycol attached to phage particles. We centrifuge, the M13 phage precipitate. After that, in the supernatant, do we want this? Do we want supernatant? Do we want supernatant? No, thank you. We discarded or out the supernatant, and after that we want to resolve the phages, resuspend phage in buffer, resuspend phage in buffer, And in phage particles, what do we have? Listen to me. We have some proteins and we have DNA. Do we want proteins? Do we want proteins? No. So, proteins, how we can add phenylchloroform, as I told you, by the portion of one phenyl and chloroform, we can add phenyl and chloroform to remove the proteins. So if we add phenyl and if we centrifuge, if we add phenyl and centrifuge, here, look at this. In the bottom side of the microtube, we have phenyl, and in the middle layer we have proteins, and in the upper side we have M13 DNA. Which part do we want? We want this. So remove aqueous layer, remove upper layer, which is aqueous. In the another clean microtube, and listen to me, I want to concentrate the DNA. How can we concentrate the DNA? As I told you, how can we concentrate it? Yes, Fatima, thank you very much. We can add ethanol. So here we can add ethanol. Which kind of ethanol? Which kind of ethanol we should use? Which kind of ethanol should we use? It should be cold. Remember, it should be cold and absolute. It means 100%. Every time we should add cold and 100% ethanol. So if we add cold 100% ethanol and centrifuge, in high speed centrifuge, we have plate of DNA. We have plate of DNA and we discard the upper side. We resuspend M13 DNA. How can we dissolve the plate? Dissolve the plate in what? So here we have DNA. We should dissolve the DNA into what? What did we add here? When we had DNA, we dissolve this with what? With listen to me again, you all know it. After we precipitate DNA with ethanol, the ethanol precipitate. We remove the upper side, let it dry, and then resuspend the plate in TE or DDW. Remember? I told you in the previous session, it is routine. Every time this repeats, okay? So, anytime, listen to me, anytime you want to precipitate the DNA, what should we use? If we want to precipitate DNA, what should we use? Ethanol. Now, listen to me. When we want to precipitate DNA, we should add ethanol. Okay? If we want to dissolve the plate, we should add DDW or TE. Understand? Okay, thank you very much. So I hope you never forget. Return it quickly. Look, at first we want to extract the M13 okay? At first we have the culture of infected cells. So we have bacteria which infected with M13, okay? If we centrifuge, because the bacteria are so heavy, they precipitate, they go down. So in plate, what do we have? In plate, we have bacteria. In supernatant, we have M13 phages. We remove it and add it to a new microtube. We want to precipitate phages. How can we precipitate phage? Because they are so light, they are so small. We should add polyethylene glycol by the side of salt. So if we add polyethylene glycol, the phage will be heavy and go down and will be precipitate. We discard the supernatant. We want this M13. Here is our M13. We resuspend M13. Now we have proteins and DNA. We want to discard the proteins. How can we discard the proteins? As I told you, as you know, we can add phenols. If we add phenol, I told you, if we can add phenol, phenol is here, proteins is here, DNA, M13 DNA is here. We remove this aqueous phase and add ethanol, cold 100% ethanol. The ethanol always precipitate DNA. Centrifuge, DNA goes down, precipitate. After that, remove the ethanol, let it dry, and dissolve the DNA into TE or DDW and can store it in freezer. Understand? Thank you. So my slide doesn't show. I don't know why. Let me refresh, please. Let me refresh. Can you see the picture? Students, can you see the picture? No? I don't know what happened. I can see the picture. So let me upload the file again, please. Let me upload the file again, please. You can see the file. Okay, thank you. Okay. Here, I just talk about some manipulation of purified DNA. So, up to now, we could extract and purify the DNA, and now we want to talk about manipulation of DNA. Okay, we want to talk about manipulation of DNA and make recombinant DNA. What is recombinant DNA? Do you know what is recombinant DNA? For example, if we add some gene, some new gene in the vectors, we have recombinant DNA. So we can make some recombinant DNA and express some genes. At first, I want to talk about some enzymes. Which we can use in manipulation of the DNA. Look at here. This is the first enzyme which we want to talk about. At first, this is Axonal Clause. Look at here. This is Axonal Clause. What does Axonal Clause do? What's its function? As you know, if we have DNA, look at here. Axonal Clause can break the nucleotide, can break the DNA from the end of the bacteries. So they can break phosphodiester bond. Between these layers of hydrogen bond, we don't want to do with hydrogen bond. The Axonal Clause break the phosphodiester. So look at here, you can see after the cleavage in both extra side we have nucleotides which break. Look at here, Axonal Clause. Do you know about Axonal Clause? How can Axonal Clause breaks the DNA? They can break from the middle of the DNA. Look at here. So Axonal Clause break DNA from its end point. Axonal Clause can break the DNA from the middle. Write this. For example, like this, like this, like this. So we have two types of nucleus. Axonal Clause and Axonal Clause. What do you mean? I didn't understand you. So, if we want to tell about some examples, for example, we have BAL31. BAL31 is a kind of axonal clause which breaks double-strand DNA. Look at here. BAL31 is a kind of axonal clause which breaks double-strand DNA. Look at here. But axonal clause 3 is a kind of axonal clause which breaks single strand. So this is double strand DNA, it breaks double strand DNA, and axonal clause 3 breaks single strand DNA. You can see it in the picture. So, please answer my question. No. I will ask you later. Let's talk about Axonal Clause 2. Look at this picture. This is S1 nucleus. S1 nucleus is type of Axonal clause, which can break single strand. You can see it, single strand. Or, for example, if we use it, we can have nick. In the DNA. For example, look at here. If we have one plasmid, how was plasmid's DNA? The plasmid's DNA was double strand circular DNA, yes? If we want to make a nick in one of the strands, which enzyme can we use? We can use S1. Yes, listen. S1 is single strand DNA Axonal clause, so it can break one strand. So if it breaks one strand, we can have nick. Okay, after using S1, it can break other strand too. What about DNase 1? DNase 1 is some kind of Axonal clause which both break single strand, look at here, which both break single strand DNA and here it can break double strand DNA. So it can both break double strand DNA. For example, if we want to break the DNA into the mononucleotide, which enzyme can be used? Can you tell me? If we want to break DNA into mononucleotides, which enzymes can be used? S1, Fatima, S1 just break one strand of DNA, single strand of DNA. But DNase 1, yes, DNase 1 breaks single strand and double strand. So if For example, in exam, I asked you which enzyme can be used for breaking DNA into mononucleotide. We can say what? Dnase 1. Okay, understand up to now? So, let's go in the previous slide. Axonal clause break from outside. We have two examples, BAL31 and Axonal clause 3. BAL31 breaks double-strand DNA from the outside. Axonal clause 3 break single-strand DNA from outside. Here, we want to talk about endonal clause. We have two examples, S1. S1 just breaks single strand. Dnase 1 breaks both single strand and double strand too. Okay? Let's talk about restriction enzyme. Do you all know about restriction enzymes? No. محدود الأثر. I don't know what do you call it in Arabic, but I think it's same. محدود الأثر. Yeah? Restriction enzyme. What do you call it in Arabic? Restriction enzyme or RE? What did you learn about it? Not just simple tacti. Not just simple tacti. It's restriction endonal clause. It should be. It should have another name. No. It should have another name. Look at here. This is tacti, okay? But this is restriction endonal clause. Restriction endonal clause, which means break in a specific site. It should have another name. Yes. So, listen to me, everyone. Restriction endonal clause can recognize the specific site in the DNA and break the specific site in the DNA. So, I repeat again. A restriction enzyme can recognize the specific site in the bacteria and can break the specific site of the DNA. تغيير, I don't know, maybe. So, a specific site. A specific site. It means, for example, I just bring some example. If we have CG, if we have this nucleotide in the DNA, for example, if we have this site in the DNA, the restriction enzyme just can recognize CG and can break the CG. Understand? So, the restriction enzyme just only recognize one site and just only break one site. Where do we use it? For example, in cloning. Do you remember? I explained it in the first session, I think. I explained it to you in the first session. I told you about cloning. Yes? We use restriction enzyme. Any questions up to now? Okay, if we have, look at here, look at my mouse here, look at my pointer. What do we have here? What do we have here? If we have any breaks or any nick in one strand of DNA, what will happen? We can have DNA ligase. DNA ligase can join two nucleotides by phosphodiester bond. So ligase can do as a glue. Do you know what is glue? Can do as glue. What does it mean? It means it can repair the nick in the DNA. So if two nucleotides phosphodiester bond breaks, it can join them together, even single strand or double strand. So it can join two molecules of DNA double strand, and it can just repair one single nucleotide break. So you all know about it. And let's go and talk about the polymerase. Let's talk about the polymerase. So we have DNA polymerase. What does DNA polymerase do? The DNA polymerase polymerases the DNA, so it can synthesize the DNA. Look at here. If we have one primer, it will attach to a template of DNA and it will continue in synthesis. So what should we want primer? Listen to me. In every DNA synthesis, we should have primer. Do anyone knows why? Do you know why do we have primer? No? Okay. In every DNA synthesis, we want separate OH. As you know, the synthesis of DNA is from 5' to 3'. It's from 5' to 3'. So in the 3', we have 3' OH. We have 3' OH should be continuously when we want to add the nucleotide. So we want primer. Why? Because we need 3' OH. Because we want 3' OH to add the new nucleotides. So every time we want to synthesize the DNA, we want primer because DNA polymerase want 3' OH to add the new nucleotide. So this is template, this is primer, new nucleotide add, new nucleotide add by the form of the template. For example, G in the front, add C, T, add A, add T, this comes. So Another DNA polymerase, it's DNA polymerase A. DNA polymerase A. DNA polymerase A, usually we can use it to fill the gaps. We call it, it can have the gap filling function. So it can gap. What does gap means? It means this gap. Okay, so DNA polymerase 1 can fill this gap. So we call it it can has the function of gap filling, and after that it can remove the other nucleotide and replace it with new nucleotides. So let me explain it more to you. DNA polymerase 1 has the process of 5' to 3' Axonal Clause. Sorry, it doesn't. 5 to 3. Which it write like this. Five to 3 Axonal Clause, and 3 to 5 Axonal Clause. Listen to me. Gap DNA polymerase 1 have the function of 5' to 3' Axonal Clause and 3' to 5' Axonal Clause. What is the function of 3' to 5' Axonal Clause? In the prokaryote cells, they can remove the primer, remove the primer 5'. Let me write it like this. Sorry. Remove the primer here is proofreading. So, what does it mean? Listen to me. This is one strand of DNA. I told you the amplification of DNA or DNA replication is from 5' to 3'. Yes. Okay. Listen to me, please. I told you it has the function of 5' to 3'. Please tell me, what is in the beginning of DNA? Always we have what? Always we have primer. Yes? Always we have primer. Understand? Do you remember? So, at the beginning of a DNA synthesis, we always have primers. So, listen to me. When I tell you it has 5' to 3' axonal clause activity, it means it removes the DNA from 5'. So, what does it remove? What does it remove? Tell me. It removes the primer. Okay. Okay. No. Now tell me if the polymerase put some wrong nucleotide. If the polymerase puts some wrong nucleotide, for example, it is A, it should add T, but it add G by fault. Okay, so this is wrong nucleotide, yes? So, if the DNA polymerase adds wrong nucleotide, what should it do? It should remove by the 3' to 5' axonal clause activity, like this. 3' to 5' axonal clause activity. They remove the wrong primer and add the right primer to. So, listen to me. DNA polymerase 1 have 2 axonal clause activity. The first one is 5' to 3'. I told you here. By the 5' to 3' axonal clause activity, it can remove primer, and by 3' to 5' axonal clause activity, it can remove a wrong primer. What does it mean? It means proofreading activity. Proofreading activity. Understand? We call it تسهیح. Yes? تسهیح. Okay. Let's which part? Look at, I told you, DNA polymerase 1, it can have the polymerase activity. It can fill the gaps. We call it, it can have the function of gap filling. For example, if there's a gap here, it can fill it. Okay? And it has the 5' to 3' axonal clause here. From 5' to 3' axonal clause. What do we have in 5'? Fatima, tell me. What do we have in 5' of DNA? The beginning of DNA. We always have primer. Thank you. So, by the function of 5' to 3' axonal clause, it can remove primer and fill it and fill the gap. Okay. So, DNA polymerase polymerize the DNA, but maybe it will bring some wrong nucleotide. For example, if this is A, it should be T, but DNA polymerase put some wrong nucleotide for, like G. What it would do, it would, by the activation of 3' to 5', remove the nucleotide from the end and add the right nucleotide and go further. We call it proofreading or تسهیح. What do you call it in Arabic? What do you call proofreading in Arabic? Yes. Yes, Tassie. So you all understand? Okay. Look at here. If this is DNA polymerase 1, if we break it into two parts, the bigger one, we call it kleno part. So, let me repeat again. If we break the DNA polymerase 1 into two parts, we call the bigger one kleno. What's the difference of kleno from DNA polymerase? Axonal Clause activity. Look at here. DNA polymerase 1 fill the gaps, but from the 5' to 3' activity, it can remove nucleotide here and replace it with new nucleotide. But keleno doesn't have the 5' to 3, 5' to 3' 5' to 3' axonal clause activity. So it doesn't have 5' to 3' activity. So it just fill the gap. But can it remove this and this? No. So if we want to just fill the gap and the enzymes doesn't remove the other nucleotides in the front of it, we can add kleno. Understand? So kleno is a part of DNA polymerase 1 enzymes which doesn't have 5' to 3' axonal clause activity. So it can just fill the gap and it can't remove the nucleotide and replace them. Okay. Let's go and talk about reverse transcriptase. What does reverse transcriptase do? Reverse transcriptase is a kind of polymerase which use RNA template. So if we have RNA template and we want to make DNA from RNA template, we can use the enzyme reverse transcriptase. What do we call DNA made from RNA template? Tell me please, what does the DNA made from RNA template? It is called as cDNA. Remember? No? I'm sure you learned about it in your bachelor degree. cDNA. You didn't study about cDNA? So, for example, if you want to amplify RNA, you can't amplify, you can't PCR RNA. You should make it to cDNA and after that amplify it. RNA. Transform it to cDNA and then amplify it, for example, PCR, real-time PCR. Real-time PCR. Did you learn about it? No? So if you don't, please search these steps for the next session because this is very important. This is very important and simple. Thank you. Do you understand up to now? There is another polymerase that I talk about is Tachypolymers. Do you know about Tachypolymers? Anybody do a PCR in the laboratory? Do you remember PCR process? Okay. Tachypolymers is the polymerase which gain from thermophile bacteria, which we use it in PCR, The bacteria is Thermophilus aquaticus. It's a very thermophile bacteria. So in PCR, because we increase the temperature to denaturate the DNA, we should use some enzymes which can tolerate the temperature, the high temperature. So we can use Tachypolymers. Okay, understand? Okay, please read it, and if you have some question, please ask me the next session. So have a great day, students. Have a great day. Please study. We have quiz next session. Okay? Thank you so much. Thank you so much. Have a great day. Thank you so much. Great day.